Hello there, my name is David Shi, and I'll be talking about our study on identifying drivers that contribute to the formation of brain metastases from lung adenocarcinoma. I'm currently at MD Anderson, though most of this work was done while I was at Dana-Farber, when the logo still looked classy. Researchers at the Broad and the Harvard were also indispensable for completing this work. Brain metastases occurred in 10% of patients with solid tumors. They represent an unmet clinical need because there are no effective treatments to date. The incidence of brain metastases are quite high, at 100,000 cases a year. The treatment for brain metastases include gamma knife, chemo, and surgical resection, and whole brain irradiation. You can imagine how the last two treatment modalities can cause extremely negative side effects on the quality of life for the patients. Brain metastases mostly derive from lung cancers and breast cancers, and today I'll be talking about lung cancers. The objective of our study is to identify drivers of brain metastases from lung adenocarcinoma with exome sequencing. We used a case-control design in which we have a case group of 73 brain metastases patients, and we compared against a control group of TCGA-LUAT patients. Details regarding the progression events for TCGA patients are not available, though based on meta-analysis and the patient's characteristics, we infer that about 30% of TCGA-LUAT patients have or will develop brain mets at some point in their lives. The contamination of our control group with true cases does reduce our statistical power, but this does not increase the false positive rate, making this comparison an appropriate one. Now, because I'm a bioinformaticist, I feel compelled to emphasize that this genomic study was anything but easy and straightforward. If the study were easy, it would not have been interesting. In fact, we have to overcome many challenges. The first of which is that brain mets are heterogeneous. Because they can come from many different primary diseases, we therefore focus the study on a single histotype, that is, lung adenocarcinoma. There's also considerable heterogeneity within a tumor, so whenever possible, we took multiple samples per patient. Varying tumor purity is also a cause for concern because it can affect our ability to detect the mutations. We therefore designed the processing pipelines to mitigate this effect. FFPE tissues also represent a real issue here. I like to drone on about how much pain formerly inflicts on us bioinformaticians, but since I'm in the job market now, I'll just say that we dealt with this as best we could. This talk will focus on copy number changes, but I also have a video posted about a tool that we developed for filtering FFPE artifacts from SME data. We deal with confounding factors using an established method that courses an exact matching. Statistical power is important, as always. Lastly, to deal with the correlation structure in the genomic data, we developed a novel statistical method that I'll talk more about later. As mentioned, tumor purity can influence detection, so we want to specifically check that our data processing pipeline is sufficient to mitigate this effect. Shown here are scatter plots of the number of copy number events against the tumor purity for both the VM-LUAT cohort and the TCGA-LUAT cohort. You can see clearly that all the trend lines are flat, and that our ability to detect copy number aberrations of different types are now strongly affected by tumor purity, and this is what we wanted to see. Next is the issue of FFPE tissues. Happily, across all the copy number aberration types, our ability to detect these changes was similar between fresh, frozen, and FFPE samples. This indicates that our pipeline is sufficiently good at removing artifacts and we can interpret our data. One important issue often overlooked in genomic studies is the issue of confounding factors. Before any matching is done, we can see that there are sex differences between our cases and controls, as well as ethnicity differences and smoking exposure differences. You can appreciate that these differences are problematic once you consider how these variables are related to the main variables of interest in the causal diagram. Our goal is to identify somatic alterations that affect the incidence of brain metastasis, but sex, ancestry, and smoking are all known to affect both somatic alterations and brain metastasis. In other words, they are confounders. In causal inference parlance, these variables introduce a backdoor path from somatic alteration to brain metastasis that is an alternative path to the direct link between somatic alterations and brain metastasis. Effectively, the differences in these confounders can mislead us into believing that specific somatic alterations can cause brain mets, even when these alterations have no effect in reality. This causal diagram also shows that age is not a confounding factor because it does not introduce a backdoor path. After coursing an exact matching to correct for sex, ancestry, and exposure, we see that there are no longer differences in these confounders between our cases and controls. This, in turn, mitigates the confounding effect. In this study, we learned a surprising lesson about power. We actually had matched primary samples for about half of the brain met patients, and we struggled for a long time with paired analysis that compared brain mets to the matched primary tumors. The reason for this became clear when we stepped back and performed power calculations on our study. In addition to the sample size number, we also need to consider whether a mutation occurs late in the metastatic progression of the tumor. In other words, whether a mutation occurred exclusively in the brain mets. Biologically, a mutation that occurs in the primary tumor can also drive brain metastasis, but we effectively disregard this type of mutation in the paired analysis. The gray vertical lines mark our sample size of 73 patients. In the optimistic scenario where most metastatic drivers occur late, we still have less than 80% power to detect drivers that are present at a prevalence of 20% or more across cases, as shown in the blue line. In contrast, the same blue line is much higher under the case control analysis, partly because we're now agnostic to whether the driver occurred early or late, and also the fact that we already have a control group of 503 patients that were already paid for by somebody else. Our workflow consists of generating copy number profiles for the case group and the control group after coarsen exact matching. We then use GStick to summarize the sample level copy number profiles into group level summary copy number profiles known as GScores. These GScores reflect the frequency of applications, deletions, as well as the magnitudes of change within each cohort. Here at the top, we have the application summary profiles for the TCGA-LUAD and BM-LUAD groups. You can see that the summary application profiles are broadly similar between the cases in red and the controls in gray. At the bottom, we have deletion summary profiles. Again, the deletion profiles are broadly similar between cases and controls. Our goal then is to identify focal changes that are different between cases and controls. Next, we took the summary copy number profiles and compared them using the Gaussian process latent difference model that we developed. After identifying the enriched amplification and deletion regions, we look for amplified and deleted genes that may drive the progression of lung adeno to brain metastasis. To give you an idea of how our Gaussian process model works, we will show some simulated data. We have case data points in yellow and controls in blue. One unique characteristic of this data is that the response variable, that is, the G-scores, do not occur at the same genomic location. For each position, we either observe the case or the control, but not both. We want to identify regions where the yellow curve is above the blue curve. In order to do so, we want to infer the vertical differences between case and control at each position. Again, we can't compute these differences directly because of the missing data for each pair of points. Therefore, we use a Gaussian process model that exploits the correlation structure along the genomic positions. Gaussian process with a double exponential kernel is just a mathematical formality for encoding the assumption that we believe that data points are nearby in position variable x should also be close by in response variable y. The model then infers for us the latent differences between cases and controls at each genomic location. We then look for regions that are above 0, which are enriched in the cases versus controls. Our Gaussian process latent difference model identified a few enriched amplification regions and one enriched deletion region in BM-LUAD versus TCGA-LUAD. Now let's zoom in into the enriched regions. First, we looked at the negative control region, NKX2-1, also known as TTNF1. This is a lineage marker for adenocarcinoma, so we do not expect it to change between brain mets and the lung adenoprimaries. And we see that the curves are on top of each other. For the enriched amplification regions, we see that one spans MEK and the other spans YAP1 and MEP13. For the enriched deletion regions, we see that CDKN2A and 2B is significantly more frequently deleted in cases versus controls. Our collaborators in Vienna then looked at the candidate amplified drivers in the external validation cohort of brain met patients using orthogonal technology. Overall, the frequency of MEK and YAP1 amplifications are much higher in this validation cohort than in the control cohort, and this provides external validation for our candidate drivers. When we first began the study, we sought out to identify differences in mutations. However, most of the known drivers of primary adenocarcinoma also observed at similar frequencies in our BM-LUAD cohort. These include KRAS, TP53, STK11, KEAP1, and EGFR. Since these genes are not observed at elevated frequencies in brain metastasis patients versus controls, these genes may not be heavily involved in the development of brain metastases. We were able to identify a few relatively unknown but plausible genes that are mutated at a higher frequency in BM-LUAD versus TCGA-LUAD. However, we had neither the resources nor the time to complete the necessary validation studies. The reviewers were also not entirely convinced by our list of candidate metastatic SME drivers. Therefore, this list of candidate SME drivers may remain in our annals of unpublished results for the foreseeable future. We were much more successful with the copy number analysis, where we identified YAP1, MMP13, and MYC as candidate drivers as you just saw. We believe that MMP13 and YAP1 might have independent roles because there are several brain met samples that only have MMP13 alterations and not YAP1 amplification. We also know that YAP1 appears to be mutually exclusive with KRAS alterations, consistent with the idea that YAP1 signaling rescues the lack of KRAS signaling in lung cancer, as previously shown by Bill Hahn's group. Next, we wanted to understand the evolutionary relationship between the brain mets and the matched primaries. In this phylogenetic tree, we can see that in this patient, MYC amplification occurs on the phylogenetic branch leading to the brain met, consistent with the notion that MYC may have contributed to the brain met formation in this patient. Another patient shows a YAP1 amplification on the branch of the brain met, suggesting that it too may have played a role in the brain met of this patient. In a more complex example, the primary and some of the brain mets actually share the YAP1 amplification, consistent with the notion that metastatic drivers can also occur early. Further, the CDKN2A, 2B deletion is unique to the brain mets 1 and 3, where brain met 3 is a recurrence of brain met 1. Importantly, we were able to validate our candidate metastatic drivers in xenograft mouse models of primary cells. This mouse work was done by Neema and Ivana. They injected primary cells overexpressing the candidates into the hearts of the mice. In this intracardial injection experiment, they observed that cells that overexpress any of the drivers more frequently metastasize to the brain than the laxity overexpression control. This increased brain met incidence was accompanied by no change in overall tumor burden. Further, they also validated that the cancer cells retained overexpression of MMP13 and MYC when they metastasize to the brain. To summarize, we conclude that MYC, MMP13, and YAP1 are bona fide drivers of brain metastasis that we identified in our case control study. In closing, I'd like to acknowledge my co-authors, institutional support, and funding sources. If you have any questions, feel free to reach out. Please use my personal email shown here because I'm now on the job market. Thank you.